Why does simple touch feel so different when the temperature shifts just a few degrees? We delved into the biological blueprint of the nervous system to discover how heat hijacks your neurons during sustained pressure. Is it comfort, or is it a neurological command your body can't ignore? External heat and pressure are raw physics, kinetic energy and thermal exchange applied against the tissue. What you perceive is an active, highly constructed internal simulation built by specialized nerve endings translating that physics into electrical data. Welcome to Blueprints of Comfort and Arousal. In this investigation, we analyze the biological responses to rhythmic pressure and thermal change. When a surface presses into your skin, your brain does not receive a single unified stream of information. It receives thousands of independent, nonlinear data points regarding texture, tissue stretch, and temperature, all arriving at different velocities. As these inputs converge, the body dynamically recalibrates its baseline neural sensitivity, actively altering how subsequent physical inputs are processed. To understand exactly how this works, we have to deconstruct the cellular matrix, tracking the signal from the superficial layers of the skin all the way up to the central processing centers in the brain. By tracing this pathway, we can decode the precise biological mechanisms that translate raw physical friction and thermal shifts into deep physiological states of comfort and arousal. The skin operates as a dense mathematical data processing matrix. Every millimeter is embedded with sensors designed to extract specific geometric and kinetic data from the environment. The peripheral hardware executing this task are mechanoreceptors. These specialized receptor organs physically distort when compressed, converting mechanical tissue deformation into electrical signals. In hairless skin, superficial SA1 and RA1 mechanoreceptors densely pack together to resolve fine textures. Deeper down, sparse SA2 and Pacinian receptors detect broad vibrations. When mechanical stress hits a threshold, they depolarize, sending a digital action potential up the type 2A nerve fibers. These specialized type 2A fibers are heavily myelinated, meaning they are wrapped in an insulating sheath. This insulation allows baseline tactile data to propagate towards the central nervous system at high velocities. Because each fiber correlates to a specific physical location on the body's surface, the brain constructs a precise, real-time spatial map based entirely on which distinct fibers are firing. Mechanical pressure maps the exact geometry and location of contact. But geometric data alone does not trigger an autonomic physical state. It requires an environmental catalyst to alter how that data is processed. That environmental catalyst is a thermal gradient. A sudden shift in surface temperature radically alters the baseline mechanical data stream being sent to the brain. This process begins at the free nerve endings, which are equipped with temperature-sensitive ion channels, known as thermo-TRPs. When exposed to a thermal gradient, these transient receptor potential channels physically open to allow ion flow. By opening these channels, external temperature acts as a biological multiplier. It merges with the pure tactile signal, amplifying and modifying the overall sensory input. The temperature of the skin directly dictates nerve conduction velocity, the speed at which electrical impulses travel along the nerve fiber. This graph charts nerve conduction velocities. Unmyelinated warm fibers travel at 1 to 2 meters per second. Conversely, myelinated cold fibers spike at 10 to 20 meters per second. This stark velocity difference exists because severe cold threatens immediate tissue damage, demanding a rapid reflex. The thermal sensory system also relies on a biological process called spatial summation, where the nervous system pools the intensity of thermal signals across the affected area. Because of this summation, applying heat to a larger patch of skin does not simply make the sensation feel wider. The aggregated neural inputs compound, causing the perceived intensity of the heat to increase exponentially. The introduction of localized heat compounds with mechanical pressure, elevating a standard tactile sensation into a high-speed, intense biological priority that commands the nervous system's attention. The thermal data stream is then compounded by a third variable, sliding, rhythmic mechanical pressure. To process this kinetic motion, the nervous system uses temporal coding. This is a decoding mechanism where the firing rates of individual afferent fibers vary over time to match the rhythmic microvibrations of the skin. The precision of this coding is absolute. Rapidly adapting mechanoreceptors fire with a spike timing accuracy down to a scale of milliseconds. This graph visualizes the temporal code. The firing rate of rapidly adapting RA1 and RA2 fibers perfectly matches the exact frequency of the rhythmic mechanical stimulus. In contrast, slowly adapting SA1 fibers maintain a flat, steady firing rate, entirely ignoring the kinetic rhythm. As this rhythm continues, the skin itself begins to change. Skin is a viscoelastic material, meaning it exhibits time-dependent mechanical behaviors like hysteresis and creep under sustained loading. During repeated cycles of stretching and compression, water in the collagen network is displaced, and elastic fibers microfragment. This physical realignment steadily decreases the skin's resistance, altering the physical dynamics of the contact over time. The sheer volume of precise temporal data from the rapidly adapting fibers, combined with the steady static pressure data, eventually floods the dorsal column medial lemniscal pathway, pushing it toward a sensory saturation point. With millions of receptors tracking the synchronized kinetic rhythm and thermal shift, the high-frequency input monopolizes the available neural bandwidth. It masks out competing environmental stimuli, forcing the brain into a state of total singular focus. Sustaining this intense level of thermal and rhythmic stimulation triggers an inevitable biological mechanism: sensory fatigue. Neurophysiologists quantify this process using the leaky integrate and fire model, which demonstrates how continuous stimulation eventually depletes a neuron's ability to generate action potentials. This graph illustrates the firing rate of a slowly adapting mechanoreceptor under static pressure. There is a massive spike upon initial contact, followed by a sharp logarithmic decay over time as the neuron's responsiveness steadily drops. The nature of the fatigue depends on the hardware. Rapidly adapting receptors shut down their firing almost immediately when a stimulus stops changing, while slowly adapting receptors undergo this gradual, continuous decay. This chart shows thermal adaptation curves. For minor temperature shifts of just plus or minus 1 degree Celsius, the skin's neural responsiveness fully resets to a flat baseline within exactly 60 seconds of continuous exposure. This decay is not a failure of the nervous system. The recovery cycle is a strict cellular necessity. Brief intervals of rest allow the displaced ions to pump back across the cell membrane, restoring the resting potential to fire again. This dynamic reset process continuously shifts the physiological boundaries. By resting and firing in sequences, the tissue avoids crossing the threshold into nociception, or pain, while maximizing the bandwidth for innocuous, pleasurable sensation. Sensory fatigue is a predictable, highly calibrated reset protocol. It protects the cellular hardware from electrical overload while ensuring the nervous system remains acutely sensitive to every new kinetic input. All of this peripheral data, the spatial geometry, the thermal acceleration, and the temporal code, must ultimately be processed by the central nervous system. This diagram shows data signals ascending the spinal thalamic tract. Pathways split, delivering sensory information to the insular cortex for conscious perception and the lateral parabrachial nucleus. The signal then extends into the lateral hypothalamus, bypassing conscious thought to initiate a deep autonomic thermoregulation response. Now, the brain's oxytocinergic system steps in as the chemical interpreter of this physical data. Oxytocin functions as a neurohormone. It actively binds to receptors in these specific brain structures and even descends into the spinal cord, directly modulating how the thermosensory and tactile pathways process the incoming signals. When rhythmic temporal coding, accelerated thermal signaling, and oxytocin release align perfectly, the result is profound. This precise convergence triggers cascading autonomic comfort and intense behavioral arousal. By defining these physical interactions through continuum mechanics and neural firing rates, we have created a mathematical map of tactile sensation. This map is the blueprint for future biomimetic haptics, allowing engineers to replicate human touch by reverse engineering the exact code of our biological hardware. Comfort and arousal are not abstract emotional states. They are mathematically precise neurological commands, hardwired into our biology and waiting for the correct physical inputs to activate. Explore the biological systems shaping our reality. Subscribe and hit the bell for our next scientific investigation beyond the boundaries of reason, only on TEMBODocs.